Sounds good. Thanks for the introduction, Jocelyn. Just to build on what Jocelyn said, I'm a PhD candidate in Dave Coltman's lab at the University of Alberta. Before this, I did my master's back in the UK at the University of Lincoln. If anyone's heard of it, congratulations. And I did my master's on glacial population genetics as well. So sheep were a bit different, but still the same basis. So I'm here today to talk to you about the spatial genetic structure of Rocky Mountain bighorn sheep at the northern portion of their range, and some work that me and a bunch of my co-authors did over the past couple of years. So by the northern portion of their range, I mean their range which extends into Canada. So primarily the range which is contained within Alberta and BC. Much of this range is contained within the national park system and within provincial parks. And it's considered a vast, pristine wilderness. And I say pristine in quotation marks because I'm very aware that it isn't as pristine and free from human interference as we might like to believe. But it's kind of the best situation we're going to get. It's a vast wilderness compared to other areas, and hence it supports many populations of species of megafauna, such as your bears, your wolves, and your mountain ungulates. So another factor which makes this area interesting is its glacial history. So around 20,000 years ago, most of Canada was covered under an ice sheet, with refugia existing in the north and the south. So the northwestern area was the Beringia refugium, and the southern area was the southern refugium. And these refugium species persisted rather than persisting in the ice areas. There were a few cryptic refugia contained within the Rocky Mountains, but these were the two main refugia. So then around 10,000 years ago, the ice sheets began to retreat and the species began to recolonize these ranges. And then we've got our current ice cover today shown on the right, which is pretty optimistic. I doubt there's actually that much ice left in the Rockies. So species within the Rockies are quite recently recolonized populations. So we should be able to see some evidence of that. So moving on to our focal species, the bighorn sheep. On the left is the distribution of bighorn sheep around the 1850s. This is the maximal possible distribution of bighorn sheep throughout Canada, America, and Mexico. And this includes your Rocky Mountain bighorn sheep, your California bighorn sheep, and your desert bighorn sheep. Fast forward to the 1960s, and as you can see, the populations have been vastly reduced. This was through the introduction of disease with European settlement and overhunting. And then if we fast forward to present day pretty much, we can see that populations are gradually recovering due to a lot of restoration and translocation efforts. But the interesting thing to note that I want to take home from this figure is that since the 1850s, almost 200 years ago, the range of bighorn sheep in Canada has never really changed. It's kind of remained the same shape at the same extent, and there's not really been much change there. So in this region, we should be able to observe the natural spatial genetic structure of Rocky Mountain bighorn sheep, versus if we were looking at populations south of the border, they've been subject to die-offs or disease and translocations, reintroductions. So we're not really getting a true picture of the natural population genetic structure if we look at it in the United States. But if we look at it here, we should get a good image of what the actual population genetic structure of Rocky Mountain bighorn sheep looks like when with minimal human disturbance or influence. So we had 4 aims for our study. The first of which was to identify the broad-scale spatial genetic structure, its causes. Then we wanted to look at the fine-scale spatial genetic structure and the extent of contemporary gene flow. We also wanted to test for sex differences in population genetic structure and gene flow across the landscape. And then finally, we wanted to look at patterns of genetic diversity that are likely to have arisen as a result of this post-glacial recolonization around 10,000 years ago. So how did we go about doing this? So we collected samples from around 50 locations throughout Alberta and British Columbia. So these sampling locations are shown as black dots on this map on the right here. As you can see, there's around 50 spread along the Rockies. In blue is the extent of the Rocky Mountain bighorn sheep's range in this area. And as you can see, we've got pretty good coverage over most of the range. I should note that the sampling density is not representative of the actual population density. So although it looks like there's more samples taken in the north, which there was, this is indicative of the sheep densities. It's not that there's more sheep in the north than there are in the south. It's just that we wanted to sample the north more heavily because this is where there's the least human disturbance, and we're likely to get a clearer picture of the natural population genetic structure of Rocky Mountain bighorn sheep. So we've got our 50 sampling locations, and what did we do with them? So from the sampling locations, we took 1,500 samples for around 1,500 individuals. Most of these samples were taken in the form of fecal samples, which were collected during the winter, just because it preserves the feces better than in the summer. Then we had quite a lot of samples that came from the Alberta Fish and Wildlife Unit, Forensics Unit. These were taken as horn core samples. And then we had some blood and tissue samples, which were taken from long-term studies such as Round Mountain, or for relocations from areas such as Lusker Greg. So from these 1,500 individuals, we genotyped all of them at 30 microsatellite markers. We did originally have a panel of around 25 to 30 microsatellite markers, but we just picked the 30 most highly variable, so the most informative markers, and genotyped individuals at these markers. We also genotyped them at one sex determining marker. This is because, for example, for the feces, we didn't really have any idea of what sex the feces came from, so we had to figure out that one way, so we used genetics to do that. From these 1,500 individuals, a 200 individuals subset was taken from around 20 locations and sequenced at a 220 base pair portion of the mitochondrial D-loop region, which was meant to be highly variable, but as you'll see later, that wasn't the case. From these 200 individuals, we obtained 188 haplotypes, which was used for further analysis. So we've got our microsatellite genotypes and our haplotypes. Next, I'm going to talk about what we did with these this genetic data. So I'm going to get into it, and from now on, everything I'm talking about until I say otherwise is using the microsatellite data. I should say it at the beginning of every slide, but I'm sure I'll forget to mention that it is. So this is all microsatellite data that we're using at the moment. So first of all, we wanted to identify the broad scale spatial genetic structure and its causes. So the first thing we did was to look at the effect of isolation by distance on genetic distance. This is common among many species. So simply we investigated the effect of Euclidean geographic distance on genetic distance. Ney's genetic distance is shown here, but all genetic distances showed the same trend. The bighorn sheep genetic distances were highly correlated with Euclidean geographic distances. In fact, 84% of genetic distance was explained by geographic distance, which is very high. That's higher than bears, wolves, deer, and elk, as far as I remember. This is likely due to the fact that bighorn sheep are habitat specialists, so their habitats are slightly more fragmented than the other species, so the occurrence of movement between habitats is slightly less than the species. But this bears the question, there's still 16% of genetic distance which is unexplained, so what explains this remaining 16%? So to investigate this remaining 16%, we used a Bayesian clustering method in structure to investigate cryptic population genetic structure. So for those of you that don't know, this is a structure plot. Each color represents a genetic cluster. There's 4 genetic clusters here. And each little bar represents an individual. These bars are tiny because there's actually 1,500 individuals in this plot, so it looks very small, but there are individual bars there. The color of each individual just represents its allocation to a genetic cluster. So we ran structure on all 1,500 individuals, and we found that there were 4 genetic clusters present, defined by the Bow River, the North Saskatchewan River, and the Athabasca River. However, James et al. Recommends that when you do structure like this, because you're doing it at such a high level, you can then go and do it at a lower hierarchical level, and it often reveals more genetic structure. So we went ahead and did that. So first of all, we looked at between the Bow River and the Athabasca River, and we found that K = 2. There were 2 genetic clusters there, which is no different from what we observed in the first place, so that kind of confirms what we observed in the first place. We then went ahead and tested from the Athabasca River northwards, and actually we found that K = 2 again. We found that the Smoky River caused an extra genetic cluster. So from our 1,500 individuals, we found 5 genetic clusters defined by the Bow River, the North Saskatchewan River, the Athabasca River, and the Smoky River. We didn't do this hierarchical analysis on anything related to the Bow River. Every time we ran structure, as soon as K = 2, as soon as we used K = 2, the Bow River was kicked out as completely different to everything north of the Bow River. So it was kind of obvious that that was a clear and definitive barrier to gene flow. So that suggests that major rivers are barriers to gene flow, and we had to investigate this in a statistical way. Oh, sorry. So this is just what I showed you back there, shown as geographically on a map. So you can see there's a genetic cluster for each inter-river region. So to test this association of rivers as barriers to gene flow, we used partial mantle tests. So for the partial mantle test, we investigated the effect of rivers on genetic distance, so whether individuals are on the same side of rivers or the opposite side of rivers on genetic distance, while controlling for Euclidean geographic distance, because we already know Euclidean geographic distance is highly influences genetic distance. So you can see these plots on the left. There are open circles represent individuals opposite sides of rivers. Closed circles represent individuals the same side of rivers, and the line represent the average. And you can see that the individuals on the opposite sides of rivers are usually above the average genetic distance than expected for their Euclidean geographic distance. This was observed for the Athabasca River, the North Saskatchewan River, and the Smoky River. However, mantle tests for the Bow River did not find the Bow River to be a significant barrier to gene flow. This is likely because there was such a vast difference between sampling locations north and south of the Bow River. So as you can see, the shortest distance between individuals separated by the Bow River was around 250 kilometers. It's unlikely bighorn sheep are going to make that migration frequently enough for the Bow River to be picked up as a barrier to gene flow. So that brings me on to my next point, which is about the maximal distance of which we can expect gene flow to occur. So to investigate fine-scale spatial genetic structure and contemporary gene flow, we perform spatial autocorrelation. So here we have relatedness on the y-axis. Here is Queller-Goodnight relatedness, but we found the same trend with all other relatedness estimators. And along the x-axis is the distance class that the pairwise relatedness belong to. So as you can see, up to around 100 kilometers, individuals are more related than average, and the average being this red line here. It's just below zero, it's negative 0.00001. So it's essentially zero. So individuals are more related than average up to around 100 kilometers, which suggests that gene flow commonly occurs up to around 100 kilometers. Now on this graph, we actually have three different classes of relatedness estimates. We've got all individuals, male individuals and female individuals. The male and the female individuals are done separately to investigate sex-biased gene flow for male and female individuals. As you can see here, there was actually no difference observed between males and females, and that's unexpected. That's not unexpected, as these microsatellites come from nuclear DNA. Hence, they're inherited from both the paternal and the maternal parties. So it's unlikely that we're going to observe any differences in gene flow here. Which brings me to my next point, which is our study of sex differences in population genetic structure and gene flow. So our analysis of spatial autocorrelation for the two different sexes found nothing, essentially. So then we went ahead and sequenced some mitochondrial DNA. So for those of you that don't know, mitochondrial DNA is maternally inherited. Hence, we should be able to look at female population genetic structure and female gene flow from this. Unfortunately, when we sequenced the 188 individuals, around 8% of the individuals had this M haplotype, this main haplotype, and the remaining 5 haplotypes that were found only differed from the M haplotype at 1 base pair. So we found a lack of variation between haplotypes and a lack of variation in the haplotypes that were actually observed in individuals. So we went ahead and plotted this on the landscape, and we found no real effect of, we couldn't really make any comment on the trends that we saw, so we had to conclude there was an absence of sex-biased dispersal. And in fact, when we compared the FSTs between mitochondrial haplotypes and nuclear genotypes, the mitochondrial haplotypes are 2 to 4 times higher than the nuclear FSTs, which is expected when there's a absence of sex-biased dispersal. So that further confirmed that. So from that, we have to confirm there's an absence of sex-biased dispersal. Finally, our final point was to characterize the fine-scale spatial patterns of genetic diversity. So to do this, we used 2 measures of genetic diversity. We used expected heterozygosity and allelic richness. We plotted both of these against latitude and looked at the trends we observed. And we observed a decline in genetic diversity moving northwards in both of these measures, which was around a 20 to 30% decline in genetic diversity between the most southern populations and the most northern populations. So we're talking from Castle Yarrow up to Narroway. So there's around a 20 to 30% decline between those populations. This is likely to do with post-glacial recolonization. So individuals recolonizing the Rockies moving northwards following the retreat of the ice sheet, in a stepping stone-like fashion, and the founder effect getting stronger and stronger with each stepping stone, essentially. So to surmise, we did observe broad-scale spatial genetic structure correlates with inter-river regions. every time I tell this, I get people saying, well, I saw a sheep crossing a river here, or these rivers aren't really barriers to sheep. They shouldn't be. I completely agree. These rivers don't actually represent physical barriers to sheep. They are, in fact, represent areas of low elevations, which are far from escaped terrain and heavily forested. So they're predator hotspots, essentially. So these are suboptimal habitats for Rocky Mountain bighorn sheep. So they avoid these areas rather than going into them. So it's not that these rivers are physical barriers, it's that they represent areas which are suboptimal for bighorn sheep. Fine-scale spatial genetic structure shows a strong pattern of isolation by distance, stronger than we've observed in bighorn sheep, in other species, and a neighborhood size of around 100 kilometers. So that means that gene flow commonly occurs up to around 100 kilometers, which is in keeping with previous telemetry data and other ecological data, which kind of makes sense. We have to conclude that we observe no sex differences in population structure or sex-biased gene flow. That's what we've got to conclude. Although it's biologically improbable, it's more likely that the males are responsible for the gene flow, but that's what we've got to conclude from our data. If anyone's got any tips on how we can pull out sex-biased gene flow from our data, then I'm all ears. We're kind of stumped. Finally, spatial patterns of genetic diversity indicate bighorn sheep did recolonize the Rockies from the Pacific Northwest following the last glacial maximum. This is in keeping with plenty of other theories and studies, so it's kind of the final nail in the coffin to say, yes, that is true, that bighorn sheep recolonized moving northwards. Our genetic data kind of confirms that. So finally, I'd like to thank people in and around the Coltman lab, as well as some other people who are responsible for providing samples or providing spatial data which was used in this presentation. Also, I'd like to thank my funders and say, if you want to read any more about this, we've got a paper out in the Canadian Journal of Zoology. And with that, I can take any questions if we've got time. And if not, you can email me questions. So thanks for listening. And does anyone have any questions?
Hey, thank you, Sam. Very interesting. We have one question so far. So Peter, which I think is Peter Newhouse probably, he's wondering if you can pick up some of the translocations that happened. So Cadiment to Ram Mountain or...
Okay, we didn't actually include any transplants for the reason that we wanted to look at the natural population genetic structure. So because we've got the pedigree for Ram Mountain, we excluded anyone who was known to be a transplant, essentially. So no, we didn't pick any of them.
We have time for one more question. Someone want to raise their hand or... So I'm wondering, are you moving to genomics to refine some of these patterns, or is that enough?
Yeah, at the minute we've got a SNP panel, which is about to run next week, which is kind of scary because it's a lot of money's worth, but it's a 50,000 SNP panel that we're running. We've already trialed a 10,000 SNP panel, and it's got pretty good results, and it's pretty user-friendly, which is nice. So we're going to use the 50,000 one next week, and we're going to use that for some fine-scale population genetics, as well as some genomics to look at changes in evolutionary changes over time, mainly at Ram Mountain.
Great. Okay, well, thanks a lot.